We're good? Everybody knows what they need to study? A lot of stuff. So what's the next chapter? Lymphatic vessels and lymphatic tissue. Similar to what vessels that we've already discussed? Veins. Yes? Similar to veins, bringing blood what? Back to the what? Back to the heart. But in this case, one of the big jobs of the lymphatic system is not to bring blood back to the heart, but to bring what back to the heart? Fluid from where? Leftover. Leftover, what? Like we cooked a big dinner on Sunday and we have leftovers? Leftover from what? What happens in the capillary beds? Exchange. Things what? From the blood vessels. And fluid. Leaves, because of that hydrostatic pressure we talked about near the end of class, last class. It's going to push out excess fluid. So where does that excess fluid have to eventually get back into? Back into the circulatory system. Is it going to have time to do that in the capillary beds of the circulatory system? No. So one of the big jobs of lymphatic vessels, and is this a two-way system or a one-way system? Yeah, well, it starts from the capillary beds. So it ends. That's probably the better way to ask that question. It's not like the circulatory system that is a continuous loop. There's an end to it, or I should say a beginning to it, that starts in the capillary beds. These are called lymphatic capillaries, and they're a little bit different than what we see in the circulatory system vessels as far as capillaries go. When I look at this diagram, which we see on page 753, at the top we see the pathway that things that are traveling in the lymphatic system are going to take during their travels while they're in there. What things are going to be in the lymphatic system as far as traveling through? Fluid, exactly. When it gets into the lymphatic system, we call that fluid, which we used to call plasma in the circulatory system. We call that fluid what? Lymph. But it's the same thing, yes? So we pick up that excess tissue fluid over in the lymphatic capillaries when there's exchange happening from the circulatory system capillaries down in the true capillary beds. Then we pick up that excess fluid and it's going to travel one way. Now, what do you think there is found in lymphatic vessels similar to veins that's going to help keep fluid traveling one way? There's valves. So we're going to see valves inside lymphatic vessels. We see those cute little valves outlined there. They kind of look like hearts in lymphatic vessels. And then along the way, the fluid is also going to pass through a series of clumps of tissue. Those clumps of tissue are called lymphatic organs or lymph nodes. Now, what's going on in the lymph node? Anybody know? Anybody ever play pinball? It's fun, isn't it? You push the ball and it goes and it bounces around in the machine until you make a mistake and it leaves the machine, right? That's kind of what a lymph node is. And fluid, as it travels up the lymphatic vessels back to the heart, where eventually all of them are going to lead, fluid is sort of going to get trapped for a little while, and it's going to bounce around in lymph nodes in lymphatic tissues. And the reason for that is surveillance. Because another very important job of the lymphatic system is surveillance. What do I mean by surveillance? Are there little policemen in those lymph nodes? There's kind of like police cells in those lymph nodes, and we've already met them. Any guess at who's hanging out in the lymph nodes waiting for foreign substances to maybe come along? No. It's called the lymphatic system. Lymphocytes! Yeah. I wonder why we call them lymphocytes. Because they hang out in the lymphatic system. Yes? So the lymphocytes that we've already met when we talked about blood, we're going to talk about in a lot more detail in the next 2 chapters. But what we're going to find distributed throughout lymph nodes and lymphatic tissue are groups of cells that are constantly monitoring fluids that are passing through. What kind of tissue, and here's a dust off the brain cells big time. What kind of tissue was the spleen and lymph nodes? It's not epithelial tissue, I'll give you a hint. That's epithelial. Nope, it's not epithelial tissue. No, it's connective tissue, someone said. Very smart. It's reticular connective tissue. What's reticular bring out in your brain? Connective tissue. Think of connective tissue. There's a whole bunch of fibers in a matrix. What was reticular fibers? They were very gentle fibers. Kind of, I described them last semester like spider webs. And in reticular connective tissue, there's tons of what? Reticular fibers, little spider webs throughout the lymph nodes and lymphatic organs. And guess what's stuck to the little spider webs? Hanging out there waiting for stuff to pass by. Lymphocytes. So lymph nodes and lymphatic tissues are going to help with surveillance as that fluid that gets collected in the capillary beds into the lymphatic system makes its way back to the circulatory system. It's going to pass through all of these little filters and kind of get bumped around just like they get bumped around in a pinball machine in each of the lymph nodes in its path. So I increase the chance that if there's something nasty in my fluids, like maybe a virus, I increase the chance that that virus is going to do what? By having them bang around in the lymph nodes, take their time getting back. Say it again. Who's talking? Oh, okay. What do I do if a virus gets in my body? What do I have to do? I have to fight it. Immune system. How am I going to know it's there? I got to bump into it. You see where I'm going with this? So if I have virus attacks my body, gets into my body fluids, and eventually makes it into my lymph fluid, as it passes through all of these little systems, what's going to happen? Eventually, it's going to bang into somebody that's going to recognize it as foreign. And that lymphocyte is then going to change. It's going to elicit an immune response or an attack. So this chapter talks about the mechanics or the tubes and how they are set up. Very short chapter. So lymphatic vessels, very similar to veins. Lymphatic tissue is lymph nodes, and larger groups of tissue, lymphoid tissue like the spleen and the thymus And you know what the thymus does for a living, right? What's it do? We learned that in the endocrine system. I said thymus. T-cell kindergarten, very good. Now we're going to figure out what happens in kindergarten in chapter 21. So, these are lymph capillaries, and they are going to allow more things to pass in quickly because they have these little flap-like mini valves at the beginning. We're going to talk about these guys again when we hit the digestive system. And everybody who's in my nutrition class knows why. What else does the lymph or lymphatic system help me do with respect to the digestive system? Well, something to do with that, right? Those big flaps are going to allow larger molecules. When I absorb lipids in the digestive system, and we're going to learn that most of the absorption of the foods that I digest are going to happen in the small intestine. When I go to bring those into blood, the lipids are still too big. So I need help. They're going to get absorbed into the lymphatic system, and then they're going to get dumped into the circulatory system. So that's another job of the lymphatic system. Excess tissue fluids, absorb lipids, help with immunity. So very, very important vessels in the lymphatic system. They're going to be widespread throughout your body. And if you look at a map of the lymphatic system, it's going to look very similar to the map of that you already kind of saw in the chapter before of what? The venous system. So it's going to follow along the same pathways, and very important collecting vessels for that excess fluid. And if you look really, really close, we see the setup for distribution of the lymphatic system. So lymph is going to be transported through these lymphatic vessels. Most of the body, and if you look at the diagram, we see drained right, drained left. Yes? The right side of the body, the right arm, everything in the right half of the thoracic region, and the right side of your head is going to be drained back into the circulatory system. Whatever fluid enters the lymphatic vessels are going to go back into the circulatory system via what? We're going to dump into an area called the right lymphatic duct into the major vein that's going to drain everything back to the heart from the top. What's that called? It's eventually going to dump into the superior vena cava. Yes? Everything else, your lower extremities, most of your abdominal pelvic region, and the left arm, left side of your head is going to drain where? Back into the circulatory system via what? The red part. It's the thoracic duct. Everybody understand that? So all of those vessels, just like the venous system, are going to grow larger and larger in size and kind of converge with each other until they dump into the right and left sides back into the circulatory system. You with me? Everything from lower is going to dump into an area called the cisterna chyli. And things that are entering that region from the digestive system are going to empty into the cisterna chyli. So that's a big kind of meeting area for lymphatic fluid. And at that point, lymphatic fluid is going to have a lot more lipids in it because it just got dumped into from where? Where were the lipids absorbed into the lymphatic system? The small intestine. And then they're going to travel up and into the thoracic duct on the right side of the body. All right, so all of these little lymph nodes and groups of lymphatic tissue are going to be part of the travel of lymph fluid. So lymph transport is mainly going to happen through a muscular pump, very similar to what we saw when we talked about return blood in the venous system. Because is there any pump in the lymphatic system? None whatsoever. So we have to rely highly on pressure changes and muscle contractions to help move that fluid back and eventually get back into the circulatory system. Sometimes things might get blocked up. Anybody ever have a lymph node that got blocked up? What happens to it? It gets all swollen. And sometimes when we're undergoing an immune response and cells are going to start to multiply during that immune response, we might get crowded in there cellular and have a swollen lymph node. So when you go to the doctors complaining of a sore throat or a headache, what's one of the first things they do? They feel up in around your neck. Why do you think they do that? Look at all those lymph nodes up and around the back of your head. Anybody have problems with those during allergy season? I swore I had brain tumors one year. The first time it happened to me, I never had problems with allergies until my 20s, which was last year. And the back of my head one spring, I woke up, my face was all swollen, and the back of my head, I could literally feel every single one of those lymph nodes. And I thought, oh, this is it, I'm a goner. But what was it? Because of the allergic reaction and the immune response that was going on, cells were developing and reproducing themselves. Fluid was backing up, especially in my head region, because of all that crowding in my lymph nodes, and caused swelling to occur. So, lymph transport in these vessels is very important. And we see branches in the thoracic region from major lymphatic trunks and ducts, all converging on each other so that they can then enter back or put back that fluid into the circulatory system. So when we have a look up close and personal at the lymphoid cells and the lymphoid tissues, again, we're mainly talking about lymphocytes. There are other cells hanging out in the lymph nodes too, but lymphocytes are going to be the major ones that we find there. Lymphocytes, if you see them here in this diagram, and this diagram is on page 756, we can see those reticular fibers I described before. Remember, lymphatic tissue is reticular connective tissue. We're going to find lots and lots of reticular fibers with tons and tons of lymphocytes stuck on them. The other type of cell, and somebody mentioned it before, we're going to find here is macrophages. What do macrophages do for a living? They're like little Pac-Men. The lymphocytes can recognize things as foreign, and how they're going to fight off foreign invaders is create a group of cells that are going to make chemicals to neutralize those foreign invaders. Foreign invaders, that's too long a word. Let's call them antigens. Okay? You've heard that word before, haven't you? What's an antigen? Not necessarily foreign. You have self-antigens. We talked about them when we talked about blood. Remember those guys? The A and the B and the RH guys. They are substances that can elicit an immune response. So an antigen can be self. And would I elicit an immune response against myself? Not normally. Sometimes this whole system goes a little haywire and you do elicit an immune response against yourself. What kind of disease is that called? It's called an autoimmune disease. That's not right. Something went wrong. So normally you don't elicit an immune response against an antigen that is a self-antigen. But all cells, all living aganoplasma membrane cells have antigens associated with them. And your cells are waiting to make sure that none of those antigens are foreign as they pass through those spaces in the reticular connective tissue. Are you with me? Okay. So, macrophages can determine that things aren't self and eat them. Lymphocytes can be much more specific in their attack. And when we talk about immune response, we're going to talk about nonspecific and specific immunity. And both of them are going to work together to keep you safe. So what we find inside the lymph node then is a pinball machine. Yes? So as fluid travels along this vast series of tubes, they're going to enter this little pinball machine through lymphatic vessels, and everybody's only going to go in one direction. So is anything ever going to go back this way out of the lymph node once it enters in? So everything's going to enter the lymph node via the afferent lymphatic vessels. And what's going to prevent them from going in the other direction? The little valves. You see what I mean by pinball machine? Once we enter, there's a bunch of little infoldings. We're going to find the reticular connective tissue and all of those cells hanging out, and little walls, just like a maze or a pinball machine, that the fluid then gets bounced around and sort of hangs out a little bit longer in a lymph node as it's traveling back to go into the circulatory system. So what we'll see specifically in a lymph node is places called germinal centers in the cortex. And we know that word, right? We have a cortex and medulla, right? We have a cortex and a medulla in these little regions as well. So in the cortical regions of lymph nodes, we find follicles, germinal centers, and then the outer regions we find all of those great reticular connective tissue and lymphatic cells hanging there, lymphocytes in particular, as fluid passes through these regions. So fluid is going to hang and bounce around these little walls that were created inside the lymph node, and those walls have a familiar name, don't they? We've heard that name before. What's a trabeculae? You've heard that before. Oh, I've heard that before. Where have I heard that before? Bone! Remember spongy bone? How they had those little tree branch looking things that formed spongy bone? They're called the same thing here inside the lymph nodes. And fluid eventually, after it bounces around inside the pinball machine for a while, and if it's got anything nasty in it, it's going to activate some of those cells, will then be passed out of the lymph node. Now, things come in faster than they leave. How come? Well, look at the picture. There's more coming in than there is going out. What do we call the lymphatic vessels that are leaving the lymph node? Efferent lymphatic vessels. And to prevent backflow, what do they have? Valves. So we see inside the lymph node itself, there are different areas where the cells are going to come in contact with anything that might be in the lymph fluid itself. So we have an afferent, efferent, in and out, and inside, and we'll look at these in lab, of course. We see parts of the lymph node where you can actually see those follicle areas and the reticular connective tissue and reticular fibers. So Those are lymph nodes, and you have thousands and thousands of lymph nodes in the pathway of lymph traveling back to the circulatory system. The book then talks about other lymphoid organs, and we call them lymph organs. Why do you think we call them lymph organs? It's because they're bigger. They're made of the same type of setup of cells with spaces for fluid to pass through, but some of them have other jobs that are going to help filter blood, for example, in the spleen. Help me get rid of some old red blood cells. So some of the other lymphatic organs are starting, usually any entrance into my body implies that whatever's coming in is probably not sterile. So we see them all around the oral region. Those things are called tonsils. When I'm breathing in, is that filtered, sterile air that you're breathing in? No, so there could possibly be foreign invaders in the air that you breathe in as well. So you have some tonsil material, larger groups of lymphatic tissue in the pharyngeal region. What's the pharyngeal region? Yeah, there's a little tube that leads from the nose down the back of your throat into the respiratory system. You share it with the digestive system, and it's called the pharynx. So that's leading in from the outside. We have to have some coverage there just in case any foreign nasty things enter our body through that opening. If we work our way down a little bit, we hit the thymus. And the thymus is T-cell kindergarten. This is where your cells, the lymphocytes that hang out in the lymphatic system, are going to learn to recognize self-antigens. They have to go to school first. We call that Immuno Competency. So in order to pass kindergarten and be set free to go hang out in the lymph nodes for the rest of your life, you have to be able to recognize self-antigens and only attack foreign antigens. If you attack self-antigens, you flunk kindergarten. And guess what happens to you? Bam! We kill you. It's a tough world. Yes? Yep. No second chances. Boom! Apoptosis for you. Okay? So, that's where cells go to become immunocompetent. And that's the proper name, not really T-cell kindergarten. We also have an area called the spleen. And in the spleen, we're going to find red pulp and white pulp. The white pulp is very similar to what we find in the lymph nodes and has a very similar job. And the red pulp is where we're going to take care of filtering blood itself from the circulatory system. So it does two jobs, that spleen. Can you live without a spleen? Yeah. Who's going to pick up the slack? If I take your spleen out, the liver is going to help me recycle red blood cells as well. And then the rest of all those other lymph nodes in my body will pick up the slack as well. So they talk a little about a splenectomy and the fact that you can live without a spleen. One of the important things, if your spleen does rupture or blood vessels rupture, there's quite a few of them that feed into the spleen. And it's very important to do what? Get it out and repair those vessels as soon as possible so you don't bleed to death. That's more important than having a spleen. The other guys can pick up the slack. So they talk about splenectomy. The other places where we're going to find groups of lymphatic tissue is dotted throughout the digestive system. Those are called Peyer's patches. And we find them in the intestine. And believe it or not, everybody thinks the appendix is useless, but that's also a mass of lymphatic tissue. Can you live without your appendix? Yes. And it's a kind of a pain in the keister because it's the entrance to the large intestine, and things sometimes get caught up in that little dangly piece of lymphatic tissue. When that happens, you might cause an infection to occur, and then we have to take it out so it doesn't do what? Burst and let out all the nasty things from your large intestine into your abdominal pelvic cavity. So taking the appendix out is more important than having the appendix at that point. Does that make sense? Okay. So the spleen, the Peyer's patches, the appendix, the thymus, and the tonsils are all what we consider lymphatic organs. So they give a little discussion about the spleen. Again, the spleen does double duty. It's not only going to help with an immune response, similar to what we saw in the lymph nodes, but we're also going to find an area called red pulp. And that is essential to filter things from the circulatory system. Very highly vascular and very important part of your lymphatic system. It's pretty good size. That's why we call it a lymphatic organ. When we dissect our pigs at the end of lab this semester, we're going to see how large the spleen is. So there's a lot of lymphocytes in the spleen because it's much larger than a lymph node. We can see different areas inside the tissue itself. The white pulp areas are very similar in structure to what we see in the lymph nodes. Lots and lots of reticular fibers, lots and lots of lymphocytes. And then the red pulp areas are going to be more concerned with taking care of filtration of the circulatory system. Same sort of setup in the thymus. Again, the thymus is where cells go to become immunocompetent. So they have to travel there before they travel to the my brain just went dead. Before they travel to the lymph nodes to hang out and wait for anything foreign to come along. Again, we see clusters of cells, reticular connective tissue. We have a medulla region and a cortex region. Tonsils, same deal. Again, we see them in several different places in the opening to the outside. So we have pharyngeal tonsils right behind your nasal cavity. Anybody have their tonsils out? And they said tonsils with adenoids. Does that ring a bell? Yeah, they're talking about all these guys. The adenoids are the ones that are up behind your nasal passages. Then we have palatine tonsils, and that's in and around what bone maybe? Palatine bone. And then we have lingual tonsils behind the tongue. So anything you put into your mouth or breathe in has some surveillance going on in that region as well. Peyer's patches in the, where are those again? Digestive system. Groups of lymphatic tissue, because everything you eat is not sterile, right? So we need Peyer's patches and the appendix to help us with foreign substances there. All mucus producing tissue has groups of cells associated with the lymphatic system, and they have a special name, MALT. M-A-L-T, and that stands for Mucosa-Associated Lymphoid Tissue. So mucous membranes leading to the outside are there to kind of hold on to anything foreign before it enters the body. We had our disgusting smoking talk, yes? There's mucus lining your trachea. Trachea is lined with pseudostratified ciliated columnar epithelial cells, right? Yeah. And in and amongst that layer, we're going to see MALT, mucosa-associated lymphoid tissue as well. You with me? Barely? That's okay. Developmental aspects, don't worry about it. We just finished chapter 20. I told you it was going to be quick. Any questions? Let's do chapter 21 now. Yeah, I told you. So chapter 21's easy. I mean, chapter 20 is easy. Chapter 21, not so easy. Chapter 21 is going to be a pain in the keister. You're welcome. I'm just giving you forewarning. See, this is the reason why I said you've got to start studying now. Because of this chapter. And this is the killer. So we are going to try to talk about the immune system as if it is a chess game. Anybody play chess? No chess? That's it? No chess player? Okay. Have you ever watched any of those movies like from oh, I can't think of the name. People in castles with moats and medieval times. Let's talk about it that way. You are the castle. And all of your little soldiers are there to protect you. You are the king or queen of the castle, and all of the soldiers are there to protect you. It's sort of like a little war or a chess game. Some of the soldiers are not very many points and kind of indispensable. Well, not indispensable, but easily. Yeah, we're not really too concerned when they do. What do they call those in the chess game? The pawns, yes? And there's a lot more of them, right? And then we have the individual, very specific players in the chess game that can do a lot more and be more specific in their attack. They're worth more points, right? Well, an immune response, the people, or the cells, I should say, that are there to protect you from foreign invaders work on the same premise. So we have something called innate defenses, kind of like the barriers that surround the castle to help keep foreign invaders out. If a foreign invader comes, they're going to bang into the wall and that's it, you're done. But sometimes those foreign invaders breach the castle walls and they can actually get in to the castle. And then who's going to fight them off? People that can recognize that they don't belong, and they can be more specific in their argument or fight, I should say, and that's called the adaptive defenses. You with me? Kinda? Anybody? All right. So this diagram shows us or gives us a general overview of what we're going to talk about very specifically in this chapter. And that is innate versus adaptive defenses or immunity. So some of the innate defenses, very non-specific, things that are there to help block any foreign substances from coming in, are things like your skin. You're wrapped up with skin, right? You also have mucous membranes. So any nasties get in your mouth or your nose, we're going to stick them on some mucus and get rid of them. So surface barriers like skin and mucous membranes are part of your very non-specific defense. It's like the walls of the castle. If I can't get over the walls of the castle, I'm not getting in, right? Once we get in, we have little sentries or soldiers around the castle wall that could maybe pick you off before you get in too deep into the castle walls. Those are also part of what we call innate or non-specific immunity. Things like our phagocytes. They grab things and eat them up and spit them out after we've destroyed them. An inflammatory response is also helping to fight off foreign substances. Remember when we talked about cutting our skin? Who squeezed out of the circulatory system and came in and ate up anything nasty that got into that cut? Who has the ability to squeeze out of the circulatory system through a process called diapedesis? Yeah, white blood cells, right? And once they get into the tissues, they're macrophages. We also have chemicals. We have kind of chemical warfare floating around in the circulatory system as well, with something called antimicrobial proteins. We have groups of proteins that can band together and poke holes in things that we'll talk about when we talk about complement. And then temperature. Sometimes when you get sick, your temperature goes up. What's that doing for me? Yeah, remember when we talked about proteins and the shape of proteins? Well, say a nasty foreign substance gets in there. It needs a certain temperature to survive. If I increase the temperature, what's going to happen to its molecular fold? It's going to unfold or denature. Problem with fever is it's also going to denature your proteins if it gets high enough. So we have to be careful with that, right? But a low-grade temperature, what did they say you should not do? Leave it alone. It's there for a reason. If you have a temperature of 99, suck it up. Don't go for the ibuprofen right away. Why do you think you have a temperature of 99? Because your body is trying to fight off that foreign substance. Go get some chicken soup and some Kool-Aid and sit on the couch and watch some SpongeBob. Okay? But yeah, well, with the starve a cold, feed a fever. Feed a fever, right? So fever is there as a non-specific or innate defense as well. And then we're going to talk about the specific stuff. Those cells, when they recognize foreign substances and how they're going to attack them and get rid of them in the body. So the beginning of the text on page 767 talks about surface barriers, skin and mucus. Did we talk at all, I know we did, but I'm just trying to jog your memory, about some of the secretions in your skin? And when we talked about skin, we talked about skin secretions, right? Did anybody remember what we called those secretions collectively? It had a certain pH on the acidic side. Do we remember our acid mantle? No? Yes? Okay. So secretions, sebum and sudoriferous gland secretions, collectively have chemicals in them that will help protect you from foreign substances. So the secretions on your skin are going to help act as bactericidal agents or antimicrobial agents. So they're toxic to bacteria. The pH alone is toxic to bacteria. Bacteria don't like acidic pH. They don't like it because it makes them blow up. That's not happy for them. Yes? So your acid mantle and secretions from your skin are going to help protect you from foreign substances. Anything leading to the outside, in women, vaginal secretions, same thing. They're very highly acidic, and they're going to help protect you from foreign substances. Stomach, say something gets into your mouth, for example, and everything you put in your mouth, is it sterile? No, so we have to kind of get rid of the bacteria before we start the digestive process. So stomach mucosa produces gastric juices. And what is the pH of gastric juices? Anybody know? Mm-hmm. No. Around 1 or 2. Because in your gastric juices, you have cells that have produced hydrochloric acid for you. So in your stomach right now, you have hydrochloric acid floating around. And remember those wonderful, nasty little critters that got trapped in your trachea? Where did they go? You had those wonderful little cilia that pushed all that nasty stuff up into your mouth, and what did you do with it? You swallow it. Who's going to help get rid of those nasty critters? Hydrochloric acid in your gastric juices. So hydrochloric acid is going to help with the digestive process, but it also is going to help protect you from foreign substances. Saliva in your mouth. When we talk about the digestive system, we're going to talk about that again. You spit all over your food, there's enzymes in there, it's going to help break down the food. But there's also chemicals in there called lysozymes. And what are they going to do for us? Lice. They're going to break down what we eat, but they're also going to break apart any nasty little critters I happen to stick in my mouth as well. So they help to destroy bacteria before they get into the digestive system. And again, don't forget about that sticky mucus. Sticky mucus is going to help trap those organisms, and usually there's going to be some sort of ciliated cell that's going to push that sticky mucus either out of the body, or in the case of the trachea, going to push it so that I swallow it and kill all of those nasty little critters in there. All right, what if they breach those defenses? What if they still get in? I have cells that are constantly on surveillance that are watching for any substances that might be foreign. So the next thing the book talks about is internal defenses, cells, and chemicals. And the first group of cells they talk about are the phagocytes. Now we already talked about this, right? So this should be repeat information. What's phagocytosis? Cell wraps itself around a foreign particle, and usually when we talk about phagocytosis, we're talking about something particulate. And in this case, they are those little green balls. Let's call the little green balls bacteria. If you look at the picture over there, that's an actual cell in the process of phagocytizing. Anybody know what those are? Those little green balls? They're bacteria. So this cell is in the process of knowing that bacteria doesn't belong and getting rid of it. Again, non-specific. I just know it doesn't belong. I know it's foreign. I know I need to get it out of there. And I need to do it as soon as possible before whatever those little green balls are get into the rest of the body. So we start with the process of phagocytosis. Oh my goodness, steps. That would make a nice essay question on your next exam. Yes? So first, phagocytes have to kind of stick to the foreign substance. And throughout the body, we're going to talk about different substances and different cells that can actually create that sticky effect of foreign invader to phagocyte. The process is called opsonization. And if you read in your book, in parentheses beside the word opsonization, anybody knows, anybody see what it says? To make tasty. Kind of like put a little salt on it. It's actually chemicals in the body that cause it to attract and stick to the phagocyte. Once it sticks to the phagocyte now has time to do what? Wrap its little plasma membrane arms around whatever the foreign substance is. So the phagocyte forms little false feet or pseudopods around the foreign substance and brings it into the cell. What's that process called when I bring something into the cell? It's called endocytosis. And it creates a package around the foreign substance. I don't want that, whatever foreign substance it is, running around my cell wreaking havoc, do I? I want to put it in a package so it doesn't cause any problems throughout the cell. That package is called a phagosome. It's just another name for packages inside the cells. What was that general term? No? Remember the term vacuole? Okay. So this one is going to happen after phagocytosis. And it's going to have a foreign little critter in there. So we call it a phagosome. All right, now that I have it in there, what do I have to do with it? Kill it. So how am I going to kill it? If I fuse it with a package of nasty chemicals called lysozymes, or lysosome is the package that contains those lysine chemicals. If I fuse the two packages, I can spill all those nasty chemicals all over the foreign little critters. And what's going to happen to them? They're going to explode or lyse. Now I have a package with nasty chemicals in it and dead little bacteria parts. That's called a phagolysosome when the two fuse. What do I do with all the dead body parts now? Throw them out via exocytosis. So exocytosis of the vesicle, that combination of the nasty chemicals and dead little bacteria body parts, are then going to leave the cell. Then where are they going to go? Just float around forever and ever? No, all that nasty stuff is then going to get filtered out of the body. Who's going to help me filter out nasty little dead bacteria body parts? My kidneys are going to help me. Right? Some of the sweat I sweat out is going to be waste products as well. You getting the point? Okay, so now it's floating in fluid and it can be distributed to the organ systems that are going to help me get rid of wastes for me in the body. So understand what phagocytes do for a living. No, they are non-specific. They just know that doesn't belong and I need to eat it. And understand what phagocytosis is. Understand the steps of phagocytosis. Damn, another one of those. The other thing they talk about in non-specific internal cells are something called a natural killer cell. And natural killer cells are a type of lymphocyte that float around in the circulatory system, and they describe them as the pit bulls of the defense system. I like that. So they're groups of large lymphocytes. Remember when we did our differential? Yoo-hoo? The differential, white blood cell differential. Okay, and you looked at the lymphocytes, and some of them were small, and some of them were a little bit larger. Is that ringing a bell? Okay, some of those larger guys might be these guys, the natural killer cells. And they are going to recognize, again, foreign substances. They're not specific like the lymphocytes we're going to meet in adaptive immunity or specific immunity. They are members of the non-specific team. So they are going to act like little cells that will go around and poke holes in things. I used to be able to describe a movie way back then, but I'm getting too old and nobody remembers the movie. But it was a really bad horror film with Karen Black in it. And there was this little, like, native guy with a spear that was possessed. And he'd run out from under the bed and poke her in the legs. I don't remember the name of the movie. I just remember it was a really bad movie. And that little guy reminds me of these natural killer cells. So they know it's foreign. They know it doesn't belong. And one of the things I can do to help get rid of foreign substances is to poke holes in them. So natural killer cells are part of our non-specific immunity. Inflammation, tissue response to injury. The inflammatory response that we talked about when we talked about the integumentary system last semester. So all of these things should be familiar to you, right? In order to increase permeability, vasodilation can occur. Permeability of what? What do we want to get out of the circulatory system and into tissues during an inflammatory response? Yeah, I want to allow those cells to squeeze out of the circulatory system through diapedesis, get into the tissues, become macrophages, and get rid of any nasty little critters that might be there when we get a cut. So this diagram on steroids talks about that, which we talked about in Chapter 5, when we get tissue injury. When we break cells open, we release cell innards. And inside the cells, there are chemicals that are going to attract these cells to the area for an immune response, for an inflammatory response. So the release of chemical mediators is what they're talking about. Things like histamines. What's a histamine? You've heard that? Granules inside a basophil? Ringing a bell? Anyone? Yep. Those histamines are also going to get called to the area in those basophils and some of the cells that contain histamine granules to the area. Another group of chemicals called complement, which we'll talk about. Kinins, prostaglandins. Again, chemicals that attract all of the cells that are going to help fight foreign invaders if they are there when I cut myself. Those chemicals are also going to cause vasodilation. Vasodilation is going to allow fluids to leave the circulatory system, along with those cells that are going to help phagocytize anything that might come in. Those chemicals will therefore increase capillary permeability. And they're also going to attract some of these guys that I just talked about to the area. Neutrophils, you know who those are? What's a neutrophil? It's one of the 5 what? Leukocytes, or white blood cells. And what did I say a neutrophil was? Begins with a P. Phagocytic. Yeah, it's a polymorphonuclear leukocyte, yes it is, but it's also a phagocytic cell. Monocytes, when they squeeze out of the circulatory system, get into the tissues. What are they now? Now they're macrophages or phagocytes. And lymphocytes are also going to come to the area, some of them non-specific, some of them specific to help fight off foreign substances. This attraction, this chemical attraction, is called chemotaxis. Anybody ever watch Batman? How does Batman know something's going on? The light in the sky goes off. Batman knows something's wrong. Where does he go? To headquarters to find out what he needs to do, right? So those chemicals are like the bat light for the cells of in an immune response. They're going to attract them to the area, chemotaxis. Leukocytes migrate to the injured area. Get out of the circulatory system, release factors, call more players to the fight. So all of these things are going to happen to help the healing process. Because of the leaking of those dilated blood vessels, more fluids leak out, and they can cause swelling. We talked about this before too, right? More pressure in the area can fire off pain receptors. Heat, redness, because of more cells and more fluid in an area to help the healing process. So we might have some temporary problems moving a joint, pain in an area where the healing process is going on. We also have clotting proteins that are going to be activated. Remember those wonderful 13 clotting factors in the clotting cascade that you learned about in blood? We're going to fire those puppies off so we can make what? Give me another name for clot. Formation of what? Platelets are going to help this process. The formation of fibrin threads from prothrombin to thrombin, fibrinogen to fibrin. Is that ringing a bell? That's what's happening here. Under clotting proteins form interstitial clots that wall off area to prevent injury to surrounding tissues. So now I'm trying to thread them back together. So temporary fibrin patches form scaffolding for repair. All of these guys are going to work together to help heal damage. Are we good? What's pus? It's all the dead soldiers. Yes? So all these guys that come to the area through phagocytosis, some of the dead tissues, some of those cells that have been there long term dying off, pus may form. Pus isn't always bad. If pus forms for too long a period, there might be an indication that there's some sort of infection that's preventing the healing process from happening. So what do we have to clear? What do we have to do to that cut? Just pile dirt in it and hope for the best? Yeah, we clear away all of that pus and dead material and let some new cells get into the area in hopes of the healing process taking place. Yes? Awesome diagram, right? So some of the things that happen during an inflammatory response: vasodilation, increased vascular permeability. That's all talked about on page 769. Some of the different chemicals involved: histamines, kinins, prostaglandins, leukotrienes, complement. All of these chemicals are going to help mobilize cells to the area and help the healing process. Phagocyte mobilization is also involved in the discussion we just had. So leukocytosis is getting leukocytes where? From the circulatory system into the tissues to fight that war. Margination, what's that? In order for me to get out, I have to do what? I got to squeeze through the cells, the endothelial cells that line a capillary bed. So margination is where the neutrophils will actually cling to the wall and then eventually squeeze their way out of the circulatory system. The squeezing my way out of the circulatory system, what's the fancy name for that? Diapedesis. So neutrophils can flatten and squeeze out of the capillary beds via diapedesis. Now, once I get into the tissues, how do I know where to go? I have to follow the bat light. What do I have to follow? The chemicals, the chemical signal. So chemotaxis is now going to help draw those neutrophils to the area, that chemical beacon, so that they can get those foreign substances and eat them up. Yes? So that's described under phagocyte mobilization in your textbook on page 771 and 773. That could possibly be an essay question as well. So on page 772, we see a table of some of the different chemicals we just described. Histamines, kinins, prostaglandins, platelet-derived growth factors. We talked a little bit about them when we talked about platelets and how they attract things to the area to help plug up clots. And then a group of chemicals called cytokines. And cytokines act to help with chemotaxis in an inflammatory response. Cytokines. I've heard that word before. Did they help cause something to happen? Maybe in chapter blood? Maybe they help to cause leukocyte differentiation from our hematopoietic stem cells. Is that ringing a bell? Yeah. So if I increase the production of this, more is going to get into the circulatory system. And who am I going to make more of to help with the war? More white blood cells. And where are they going to come from? The bone marrow. Hematopoietic stem cells, or hemocytoblasts, are going to be stimulated to go into the maturation series for a leukocyte by groups of chemicals called cytokines. Yes? Is your brain ready to explode yet? Oh gosh, there's so much more. So, summary, again, our wonderful tables in our book. We want to take advantage of them in this chapter for sure. So we have summary of innate body defenses. So we see a general description of our epidermis, and everybody knows that's the outside layer of your skin. Skin's made of dermis and epidermis. Remember the cells of Langerhans? Is that ringing a bell? We found them in the layers of the epidermis when we talked about the 5 layers of the epidermis. What were they? I'll give you a hint. We're on the immune system. The Langerhans cells are part of what? Yeah, these guys, your immune response. They're going to help monitor things going on in the epidermis. Your acid mantle is also very important. The chemicals secreted by your sudoriferous and sebaceous glands. Remember those guys? The oil-producing and the sweat-producing glands in your epidermis and dermis. More, we find them in the dermis. Keratin, what's that? That's your waterproofer. Guess what else it's going to help with? Bactericidal agents. So it's going to help get rid of nasty little bacteria as well. Mucous membranes, nasal hairs, cilia, gastric juices, acid mantle of the vagina, lacrimal secretions. Your tears have bactericidal agents in them. Because the eye is exposed to what? The outside, nasty, full of germs world. Urine. Urine. It's also a bactericidal agent. Urine, under normal circumstances, is sterile. This is why, ladies, I'm going to give you a little talk. In the ladies' room. Sit down! Don't pee all over the seat. If you sprinkle when you tinkle, please be neat and wipe the seat. But do not worry, ladies who hover, because urine is sterile. And if you hit the hole in the middle, the urine is going to go in the water, right? So sit down. Urine is sterile. When it passes, no, I don't even hear that. When it passes out the urethra, it's going to help to clear the urethra. So and I'm directing this at women because women have a very short urethra. The place where we get from the holding tank, the bladder, out of the body. It's a very short tube. Men, your tube's longer. You don't tend to get as many urinary tract infections. Okay? One of the things that women tend to do is, oh, I have to pee. I'm busy. Right? And they hold it and they wait. And the sensation will go away for a while. But guess what happens? Who's ever marching their way up your tubes can make it further. When you urinate, you do what? You flush them and you clear them. So don't hold your urine, ladies. Pee when you have to pee, because that's going to help with the whole urinary tract thing as well. Short urethra. So urine, very important first line of defense. Second line of defense, and again, still non-specific. We have our phagocytes, our natural killer cells, the inflammatory response, antimicrobial proteins. They describe a group of proteins called interferons. We're going to talk about those next class. A group of proteins that can band together and wreak havoc with foreign invaders called complement. And again, fever is also a help. So we'll pick up on Thursday with antimicrobial proteins on page 773.